Welcome to the T-cell mediated immunity module. In this talk, we're going to learn about how naive T cells are able to enter secondary lymphoid tissues. We will first learn about the role of chemokines in attracting T cells to lymphoid tissues, followed by the steps involved in the T cell entry into these lymphoid tissues. Once they have entered, we learn about how they leave the lymphoid tissues. B and T cells are formed in primary lymphoid organs like the thymus and the bone marrow. Even though B and T cells play an important role in adaptive immune responses, the adaptive immune responses are actually initiated in the secondary lymphoid organs and not the primary lymphoid organs. Thus, adaptive immune responses are initiated in lymph nodes, in the spleen, or in mucosa-associated lymphoid tissues. Thus, the B and T cells have to go to the secondary lymphoid organs in order to be activated. B cells and T cells are organized in specific ways in the secondary lymphoid organs, and thus in each of the secondary lymphoid organs or tissues, we are able to observe B cell zones and T cell zones. B and T cells come to the secondary lymphoid tissues together from the blood. So once they enter the secondary lymphoid tissues, how do they know where the B cell zone is and where the T cell zone is? So this is where chemokines come into play, and they help the cells go to their appropriate zones. So let us look at this figure. And in this figure, we're looking at the lymph node. And in the lymph node, there are these stromal cells. And these stromal cells, they secrete a cytokine which functions as a chemokine called CCL21. Now this CCL21 can be sensed by a receptor called CCR7. So T cells express the receptor CCR7. In addition to T cells, dendritic cells also express the receptor for CCL21, which is CCR7. And thus, dendritic cells come to the lymph node also. Now, once the dendritic cells are in the lymph node, they in turn are able to secrete another chemokine called CCL19. CCL19 is also recognized or sensed by CCR7. And thus, the T cells are able to follow the trail of CCL21 and CCL19, and that is how they're able to go into the lymph node and they stay in their T cell zones. Now let us see what B cells do and how do they know where to go. So B cells also express CCR7, and hence they're coming into the high endothelial venules and they enter the lymph node. Now, B cells also express another chemokine receptor called CXCR5. And this CXCR5 plays a role because it is able to sense a chemokine called CXCL13. Now, when we look at the lymph node, in the lymphoid follicles, we have these follicular dendritic cells. And these follicular dendritic cells make the chemokine CXCL13. So once the B cells are able to enter the lymph node by following the trail of CCL21 through the actions of CCR7, once they're in the lymph node, they're able to go to the lymphoid follicle because they're able to sense the chemokine CXCL13 made by the follicular dendritic cells. And they're able to sense CXCL13 because they are expressing the receptor called CXCR5. And so this way, the B cells and the T cells are able to determine where they have to be in the lymph node. Let us now look at how the T cells are able to enter the lymph node through the blood. So remember, they are in the blood, these high endothelial venules, and they have to leave that blood vessel to enter the lymph node. So how does that happen? And that process is very, very similar to the process of extravasation that we learned for neutrophils and monocytes. Now, T cells express the molecules L-selectins, and that is what we can see in the figure. The L-selectins are able to bind the Siala-Lewis X moiety. So on the endothelial cells of that venule, we can see these proteins called addresins, which are adhesion molecules. And on these adhesion molecules, we have the sialyl-Lewis X moiety. So the L-selectin on the T lymphocyte is able to interact with the sialyl-Lewis X moiety of the addressin glycan 1. This interaction promotes rolling, and thus the T lymphocyte starts slowing down as it moves through that endothelial venule. Now, in that endothelial venule, we do see CCL21 being made. Now, this CCL21, as we saw before, is made by these stromal cells that are present in the lymph node. So this CCL21 that is present is able to bind to the CCR7 on the T cell. This activates a signaling pathway in the T cell, and what happens is it triggers a conformational change in an integrin present on the T cell. And this conformational change results in that integrin interacting with another adhesion molecule called as ICAM that's present on the endothelial cell. So this basically results in a really tight binding between the T cell and the endothelial cell through the interactions of that integrin and the ICAM. So here we can see the process of adhesion or tight binding. So once the process of rolling and tight adhesion occurs, the next process is diapedesis. In diapedesis, the T cells will leave the blood stream or the blood vessel or the venule in this case, and they will be able to enter the lymph node. Now, as shown here, the way the T cell is able to leave the venule is by squeezing through the junctions between the endothelial cells. Now, the T cells are able to move towards the T cell zone once they enter that lymph node because of the presence of those chemokines CCL19 and CCL21 that we talked about before. Now, the T cell remains in the T cell zone and it's able to scan the dendritic cell surfaces that are present in that lymph node, and they're basically scanning for peptide MHC complexes that the TCR can bind to. Now, if a T cell is able to find its specific peptide MHC complex, then it will stay in the lymph node. If the T cell cannot find its specific peptide MHC complex, then the T cell will leave or exit the lymph node. And for that, it basically has to find the efferent lymphatic vessel. And so it leaves the lymph node through the efferent lymphatic vessel. Now, T cells express the S1P receptor, and as we know, the S1P receptor binds to the ligand S1P, which is also called as sphingosine 1-phosphate. The concentration of S1P is low in the lymph node. However, there is a higher concentration in the efferent lymphatic vessel, and this concentration gradient allows the T cell to be able to find the efferent lymphatic vessel and leave the lymph node. Now, if the T cell is able to find the specific peptide MHC complex that binds to its T cell receptor, what will happen is, as we can see in this figure, it forms an interaction with that antigen-presenting cell. So here we can see a T lymphocyte whose T cell receptor has been interacting with the peptide MHC complex with the antigen-presenting cell, which is a dendritic cell. In this scenario, that T lymphocyte has to stay in the lymph node and should not leave. So one way it is able to achieve that is that it decreases or reduces the expression of the S1P receptor. Once it has reduced the expression of S1P receptor, it will not follow the trail of S1P to leave the lymph node. Then that T cell is undergo activation and it will form effector T cells. Now these effector T cells, once they are formed, they now have to leave the lymph node. And so they will start upregulating the expression of the S1P receptor. With this upregulation of the S1P receptor, these effector T cells now can follow the trail of S1P and they are able to exit the endothelial cells that are forming the lymphatic vessel, and they're not able to enter into the lymphatic vessel and move around that way. With this, we come to the end of our talk, where we learned about the role of chemokines in helping T cells to come to lymphoid tissues and the steps involved in T cell entry into the lymphoid tissues. We also learned about how T cells are able to leave lymphoid tissues.